So at this point, up to this point, we've talked about these three different modes of selection. Selection that can be stabilizing, selection that is directional, and selection that is disruptive. One thing to note when we're talking about the kind of shifts that we see in populations, selection doesn't have to be kind of that all or nothing thing. It doesn't have to be life and death or no reproduction versus copious reproduction. For humans, it would only take about 10,000 years for a trait with a 1% disadvantage to change from 99% frequency to less than a 1% frequency, as long as that selection is consistent and unidirectional. And so we do have some traits that have emerged in less than the past 10,000 years. One of those is lactose tolerance as an adult. All infants can digest lactose because mother's milk has lactose as one of its milk sugars. But in general, after we're weaned, we lose that ability to digest lactose. The gene for lactose tolerance only emerged roughly 6,000 years ago with herding populations in the Middle East, and then spread into Europe as those agriculturalists then were able to displace the hunters and gatherers that were still living across Europe. So we do still see evidence of biological evolution, of genetic evolution among human populations. Even seemingly trivial traits can be associated with differential reproductive success. For example, there is one trait called the cephalic index, which essentially is just the degree to which someone's head is round. And so it kind of it runs on a distribution of like a cone head from the old Saturday Night Live series to Stewie from Family Guy or Hey Arnold with that kind of football shaped head. In general, having a rounder head is associated with higher survivorship, but we would think of something like malformations in the head as being purely cosmetic versus having any kind of carryover impact on reproduction. And no trait is adaptive in all environments, and really human brain size is one of the best examples of this, that we wouldn't have been able to innovate a brain of 1350 cubic centimeters 5 million years ago. It's simply what we didn't have the diet, we didn't have the technology, we didn't have the culture to be able to support that. And so traits like large brains, if things were to very rapidly shift to the point that we could no longer support the metabolic cost of our incredibly large brain, then we would find that having a large brain would be maladaptive. So you've got to have the environment favor these traits in order for those traits to be kept around. And so looking at some of the various human traits, some are very old, like the chemical machinery of the cell that comes from the first eukaryotes or true cells. Vertebrate physiology comes from obviously the first vertebrates. Face-to-face group of kin, friends, etc., dates to non-human primates, where we started seeing larger group sizes. The apparatus associated with technology dates to about 5 million years ago, as hominins emerged on the scene with Ardipithecus and Australopithecus. The mental apparatus associated with human technology or lithic technology, these stone tools. Weenie! 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 The mental apparatus that's associated with human technology. This is the advent of stone tools, lithic technology. This dates back to roughly 5 million years ago with hominins moving out on the African savanna with Ardipithecus and Australopithecus. And then the mental apparatus that's associated with human language. We're not exactly sure when language emerged, but it was probably roughly 2 million years ago. I mean, the cultural innovations that Homo erectus was able to create really lend to that greater cognition. And so it though language for Homo erectus would have sounded very different than modern human language, I think it's fair to say that Homo erectus would have at least possessed kind of a rudimentary language. So, how then, if we've talked about these different modes of selection, I mean, it's easy to think about how something like lactose tolerance would be beneficial for humans, or larger brains would be beneficial for humans, um, or the capacity for language. But how then do we inherit behaviors? How does heritability apply to behaviors? So, one of the biggest problems in accepting an evolutionary view of behavioral variation is this question of how heritability translates into phenotype. People who are more on the nurture end of this nature versus nurture debate would say that behavioral variation can't be heritable because so much of it is socially learned, cultural in nature, flexible in its expression, and contingent upon conditions that change from moment to moment. So how then can evolutionary biology be relevant? Well, we're going to talk briefly about the mechanisms of heritability, specifically with Mendelian genetics, and we'll certainly give a nod to the fact that most of our genetics is not as simple as Mendelian genetics, but this is not a genetics class, so we're not going into all of the mechanisms of genetic heritability, that we will briefly touch on some non-genetic mechanisms like epigenetic factors and such. But we are going to look then, after we have this rudimentary understanding of genetics, we are going to look at some animal cases of behavior to determine whether behavior can evolve. Because presumably, if we're able to see the evolution of behavior among animals, we should also be able to see the evolution of behavior among humans. And so Mendelian genetics is really our starting point. Gregor Mendel lived from 1822 to 1884. He was a monk. He's the father of modern day genetics, particularly, of course, Mendelian genetics. So he was from Austria, and in his gardening work, particularly with pea plants, he discovered the principles of heredity. So he was looking at traits of pea plants and determined dominance and recessiveness, determined that traits are carried on discrete particles called genes. And so Gregor Mendel studied 7 characteristics of peas, including whether they were round or wrinkled, yellow or green, purple or white petals of the flowers, inflated or pinched pea pods, green or yellow pea pods, axial or terminal flowers, or long or short stems. And he really got lucky because each of these traits in pea plants is a situation of one gene, one trait. So by altering the allele, you see a very obvious effect on the overall phenotype. So you don't have to worry about sequencing DNA or doing anything such as like polymerase chain reaction or any kind of genetic work. What he found was that when you crossed what we call purebred parent generation, so pure yellow with pure green in the F0 generation, you get an F1 or first offspring generation that's all yellow. What this demonstrates, because we know the difference between genotype and phenotype, the genotype of that first generation, they're all going to be what we call heterozygous. That is, they're going to have one allele for green and one allele for yellow. This recessiveness of the green isn't going to come or isn't going to be visible until your F2 generation, the grand offspring of that first cross, when you will have one phenotypically green pea pod and 3 phenotypically yellows. And so Mendel's explanation involved Punnett squares, and you guys have probably done this in high school biology, that the idea that each allele within a cell will independently assort, and from your parents you will get one allele from your mother, one allele from your father, and that will then comprise your genotype. And then when we have dominance and recessiveness here, yellow is dominant over green. The phenotype of that F1 generation is going to be dominant, though the phenotype or the genotype is not. The genotype is heterozygous with that recessive allele being masked. When you cross the F1 generation to get your F2 generation, you're crossing two heterozygotes, so they contribute one dominant allele each and one recessive allele each. What that means is that we have a ratio of genotypes in the offspring generation of 1 to 2 to 1. That is, we have one purebred yellow, one purebred green, and two heterozygous yellow. If we look at the ratio of phenotypes, however, it's 3 to 1. It's 3 yellow phenotypes, 1 green phenotype. So we already, just with Mendelian genetics, with his pea plants, see that genotype doesn't necessarily 100% determine phenotype. What does that mean? That means that in this case of dominance and recessiveness, we can't determine the genotype of this F2 generation just by looking at their phenotype, because there are multiple genotypes that can yield a yellow phenotype. Then Mendel looked at two traits together. We can draw our Punnett squares the same way, allocating your A alleles and your B alleles, and we get this ratio then of phenotypes of 9 to 3 to 3 to 1. So we have the discrete kind of inheritance of each allele separately. That means they aren't linked. That means that the color of the seed and the rounded or wrinkledness of the seed is not linked. They're inherited independently. So what Mendel concluded was that there are two alleles for each trait, that those alleles, parents contribute only one of them to their offspring, that those alleles are contributed with equal probability. That is, parents aren't able to do anything to ensure that they're passing a certain allele on to their offspring. We have independent assortment. The alleles for different traits segregate independently. So there's not linkage so that every yellow seed is also wrinkled. That the yellow versus green alleles are independent of the wrinkled versus round alleles. 1 through 3 are the principles of segregation, and 4 is the principle of independent assortment. So we have, we segregate the alleles from our parents and their gametes. We inherit one of those. Traits are inherited independently. There are, of course, some notable ways that we don't see this strict kind of Mendelian genetics. One of those is with incomplete dominance, where we don't have pure dominance and recessiveness. What this means is that those individuals who are heterozygous, who have one of each allele, will display an intermediate phenotype. So here's an example with snapdragons, where you've got the phenotype for either red or white flowers. The F1 generation is all pink because they've got one red allele and one light white allele, so it kind of blends those two colors together. We also see this present in some diseases like sickle cell anemia. The reason we haven't gotten rid of sickle cell anemia is because individuals who are heterozygous actually confer a genetic advantage in terms of innate immunity to malaria. But individuals who are heterozygous will experience intermediate sickling when their blood cells are exposed to really low oxygen. So if they went from sea level Africa up to the top of Mount Kilimanjaro, they might have a little bit of trouble breathing because their blood cells are going to sickle slightly. Now, of course, being purebred for or homozygous for sickle cell anemia, you have evidence of the disease and you may not even live to adulthood. So we see this incomplete dominance, however, in this heterozygous condition. We can also have codominance. This is where you get the full phenotypic expression of both alleles in the heterozygous condition. So here we've got a gene for red fur or red hair, white hair. When you mix them together, you get something intermediate that we call a roan horse, where you've got hairs that are red and hairs that are white. We have also seen something similar in some populations of bees where you may have an allele for horizontal stripes and an allele for vertical stripes. And if you're heterozygous, you have both horizontal and vertical stripes and thusly appear black. So our genotype is defined as the genetic makeup of the individual. This is includes the actual genes coded for by an individual's DNA. If you were to sequence your DNA, you'd get output that said something like CCTAGUGU, so on and so forth. And that would then be able to be read by individuals who know what the biochemical makeup is of different alleles to determine what genes you possess. But really, this is not what natural selection acts on. During meiosis or gamete formation, this genetic material is copied and enclosed within the gametes, though of course it's only half of the parent's genetic information. So given perfect replication, perfect DNA copying, this genetic material is passed on to subsequent generations identically. There shouldn't be changes. Though variation in genomes is one of the inherent benefits, really, of sexual recombination. The whole reason that we engage in sexual reproduction is to introduce variability into our population. Well, of course, we know that we don't always have perfect DNA copying, and so there are other ways that we introduce variation as well, one of those being mutation. So mutation refers to the imperfect copying of DNA strands. We have a general baseline mutation rate. That is about 10 to the minus 4 to 10 to the minus 6 per gene per generation. We've got about 30,000 genes. That means that you will have roughly one mutation per every 3 offspring born. Well, we're not turning into the X-Men, right? We're not mutants that have these phenomenal kind of superpowers, so why not? There's a lot of redundance in the genetic code. So when the DNA unzips and the RNA comes in to copy it, the RNA then travels off to the ribosomes to translate that into another strand. And it's organized then through what we call codons. So every 3 base pairs of your DNA encodes for a codon, which, or when grouped together by 3, codes for an amino acid. There is redundance in the way that codons are translated into amino acids. So what that means is that you can have a mutation or you can change one of your base pairs. And if that base pair happens to be the middle base pair, you're not going to change the resultant amino acid. That means that CAG and CCG may code for the exact same amino acid. So you would have a mutation, yes, but the effects of that mutation would be functionally neutral because it's not going to influence the amino acid. We also then, once we have amino acids, we have to string those together to form proteins, and there's redundance in this as well. And so let's say you have tyrosine, lysine, and then you have tyrosine, lysine in terms of your amino acids. You're not necessarily going to get a different protein. So you can change one base pair and not change your amino acid. You can change your amino acid and not change the structure or the function of that. Well, you could change the structure slightly, but you won't change the function of that final protein that's produced. And so what this means is that most of the mutations that we experience are functionally neutral. They don't have an effect on our metabolism, our morphology, etc. They don't contribute to increased survival or decreased survival. So we can generally tend to ignore most mutations. Now, the exception here, of course, is if those mutations result in changing the pattern of gene expression. So there are certain codons that code for starting and stopping translation. And so if we change the expression of the gene, if we start copying it at a different point, or we stop copying it short of the intended end because we've had a mutation that affects the start and stop, then we get something that is functionally very, very different. But in general, mutations aren't really that worrisome. Most of them are going to be functionally neutral. We also get key critical variation through recombination. This is putting together new and novel combinations of genetic material through the act of sexual reproduction. So, of course, with recombination, your dad contributes half of your alleles, your mom contributes half of your alleles, and depending on whether they come from the same population or not, how closely related they are or not, you may end up with new combinations of alleles that aren't present yet in the breeding population. So what's the benefit of this? The benefit is that this is really important variation that can pay off when environments are rapidly changing. And so we'll come back and visit this topic when we talk about anisogamy, which is unequal gamete sizes, and we'll talk about the evolution of two sexes. But in general, when environments are changing, you have higher fitness if you reproduce sexually than if you reproduce asexually, because you can't predict what that future environment is, and so you can't predict what combinations of alleles may then pay off in that future environment. We also see variability introduced from crossing over. This is where chromosomes, when they line up in the center of the nucleus to be copied, where they might exchange little pieces of themselves. So the chromosome from your mom, as it lines up to divide into these gamete cells, you might, from the, like, let's say left half, or the left chromosome and the right chromosome, because of course you have pairs of chromosomes in your adult body, they may not separate entirely as they're lining up, and so a bit from the right chromosome may transpose onto the left chromosome and introduce an allelic combination of genes that wasn't present before that particular meiotic event. How then does genotype translate into phenotype? Genotype, remember, is your genetic makeup. Phenotype is defined as the observed expression of a trait. You guys need to make sure you know these two definitions, because these will absolutely, you'll have to discuss the distinction between the two on that very first exam. So some traits are coded for directly by genotype. For some traits, the expression of the phenotype is very closely tied to the genotype. For example, blood type, eye color, etc. You're not going to have blue eyes if you don't have an allele for blue eyes, right? You're not going to have type AB blood if you don't have one allele that's A and one allele that's B. Many traits, however, are very strongly influenced by the environment. So, for example, height and weight both have a very strong genetic component, but one of these you are able to alter more readily than the other. And so we, of course, all know that changing how much we weigh is a bit easier than changing how tall we are. But even with respect to height, that's influenced by environment as well. There have been some studies of identical twins that were reared in separate environments. So they were adopted, and one was raised in a wealthy family, one was raised in a poor family. And what you find is that the identical twin that's raised in a poor family has smaller overall stature. So your exposure to appropriate calories growing up while you are growing your adult body height can have a marked effect on your overall achieved adult body height.